So we are back, and we're going to talk about the difference between inorganic and organic molecules, and then we're going to talk about the organic molecules that make up living things. Anything that is inorganic, typically we assume it doesn't have carbon. Very few inorganic molecules contain carbon. One example, or I'll give you two examples of inorganic molecules that do contain carbon: carbon dioxide and carbon monoxide. But other than those, we really don't have inorganic molecules that contain carbon. Organic molecules always contain carbon, and they contain hydrogen. Inorganic molecules are very simple in general, whereas organic molecules are complex. They can be very large and complex. So organic molecules can form huge structures. These large structures are called macromolecules or polymers. And they're made up of multiple smaller subunits called monomers. So monomers make up polymers. And they're linked together through a process called dehydration synthesis. So in the word dehydration synthesis, or in the phrase dehydration synthesis, you already can figure that water must be being removed, because if you are dehydrating something, you're removing water. So in this reaction, we take water away and we bond the molecules together, the monomers together. We can break the link then using a hydrolysis reaction. Hydrolysis is breaking down by the use of water. So we add water, water goes into the system and breaks the monomers back to their individual subunits. And that's what I just said here. So dehydration synthesis, or a dehydration reaction. You're going to take water out and you produce a bond. In a hydrolysis reaction, we add water in and we break a bond. So here you have some monomer. This could be glucose on this side, and this monomer here could be fructose. When we take the water away and we produce this bond, we produce this dimer or disaccharide if we have glucose and fructose, and that would be sucrose. And do you guys all know what sucrose is? So I'm looking at all my stuffed animals that I keep on my table in my mini office basement area, and none of them are answering me, which is not surprising, since they're not going to talk to me in general. Anyways, sucrose is table sugar. So it's what you put on your cornflakes, or what you put in your coffee if you like sugar in your coffee. We can break them back down into their monomers if we add water back. So that's hydrolysis. So the four macromolecules that make up living things are carbohydrates, proteins, nucleic acids, and fats or lipids. And we're going to talk about each of them right now. Carbohydrates are the first of the macromolecules, and carbohydrates contain a carbon, hydrogen, and oxygen at a 1:2:1 ratio. So typically, you're going to have an example that most of you will have heard at some point, and if not, you hear it here now. Glucose is made up of carbon, hydrogen, and oxygen. The chemical formula of glucose is C6H12O6. So you have 6 carbons, 12 hydrogens, 6 oxygens. So a 1:2:1 ratio of carbon to hydrogen to oxygen. Carbohydrates make up your starches and your sugars. And sugar and starch is what gives us our primary energy for animals. And it also gives energy to plants, but it also produces part of the building blocks of the plant structure. So the smallest of your carbohydrates are your monosaccharides. These are the monomers that can make up larger polysaccharides. Monosaccharides have between 3 and 7 carbons, and they tend to form either rings or chains. So here are examples of monosaccharides. Glucose and fructose are both monosaccharides. Another monosaccharide is galactose. It's not shown here though. Glucose and fructose have the exact same chemical formula, and you can see that down here, C6H12O6, but they have a different makeup. So the atoms are not in the same exact order. And because of that, they're called isomers of each other. And the difference in the structure actually makes fructose sweeter than glucose. So disaccharides are formed when monosaccharides come together through dehydration synthesis. And a disaccharide is just 2 simple sugars, like glucose and glucose, or glucose and fructose, forming the disaccharides. The disaccharides that we often talk about are sucrose, which is the sugar that we use in our cooking, lactose, which is made up of glucose and galactose, and maltose, which is glucose and glucose, and that is cane sugar. So lactose intolerance is the inability to break down lactose back into its glucose and galactose monomers. And you've all heard of people who are lactose intolerant. They don't have an enzyme that functions properly. That enzyme would be called lactase. So polysaccharides are very large macromolecules, or the polymers that make up your carbohydrates. Polysaccharides are ways that we can store our simple sugars when we don't need as much. And they also can act as fiber in our diet. So starch is an example of a polysaccharide that we use to gain nutrients. So we eat potatoes or other molecules, or other foods that contain starch, and we gain that starch. In our cells, we hold or carry polysaccharides in the form of glycogen. Glycogen is our storage form of the large polysaccharides when we don't need as much sugar in our blood. So think about when you eat a large meal and you get a lot of sugar in your system. Say you ate 3 jelly donuts. You get a lot of sugar. We don't need all that sugar in our blood. We're going to pull out the sugar, make up glycogen that will be stored in our muscles and in our liver. And then when we need the glycogen, we can break it down for energy. We also have another type of polysaccharide that we use in our diets, and it's called cellulose. This is a carbohydrate that we cannot break down. Our bodies can't break down cellulose, so we use this cellulose as the fiber in our diets. And so there is what starch looks like. And you see that's from a potato, and you can see the starch granules inside of the potato. There's glycogen. Glycogen is found in our liver as well as in muscles, and it's our storage form of glucose, so that we have glucose available when we need it. And then there's the cellulose that our bodies cannot break down. Cellulose makes up plant cell walls. So it's very structural, and it goes right through our body, and then it helps to push out other materials in our intestines so that we get rid of all the waste. The next macromolecule that we'll talk about are the lipids or fats. Lipids are organic molecules. They also contain carbon, hydrogen, and oxygen, but not in a 1 to 2 to 1 ratio. Typically, we have a lot more carbons and hydrogens than we do oxygens. Lipids are nonpolar, so that means they will not dissolve in water. They're called hydrophobic. Have you ever heard that term before? You can think of hydro is water. Phobic means scared of. So if you have a phobia, you're scared of something, fear of. So lipids are water-hating molecules. They do not interact well with water. Triglycerides are fats that are taken in when we eat either plant or animal foods. If it's triglycerides from plants, it's usually a liquid form. If it's a triglyceride from animals, it's usually a solid form at room temperature. Triglycerides are for stored energy. And so here is what a triglyceride looks like. It has a polar region up here, and then it has non-polar region back here. So it's called an amphipathic molecule. Amphipathic means it has two different charges. The polar region will interact with water. The non-polar region will not. Oh, sorry. There we go. That's what we were looking at. So here we can see triglycerides in plants. Plant triglycerides tend to be liquid at room temperature. And so here you can see the structure. They tend to have double bonds in the fatty acid chains. And so this would be considered an unsaturated fat, whereas triglycerides in animal form tend to be saturated. And so when they're saturated, they tend to be solid at room temperature. The unsaturated, I'm going to go back for a second, the unsaturated fats can have one double bond or can have more than one double bond. So you've heard of monounsaturated fats versus polyunsaturated fats. Polyunsaturated fats means many unsaturated regions or many double bonds, okay? And we've all heard of trans fats. You know that, or I don't know if you know this, so I guess I'm telling you this now, and then you'll know. Most people know that animal fat is typically considered more unhealthy than plant fat. So if you're eating a lot of fatty foods that are associated with animal fat, it's going to give you a lot more cholesterol, a lot more fat in your system that could potentially clog your arteries. Unsaturated fats, so plant fats, tend to be healthier fats. I'm drinking my coffee right now without sugar. I just do my cream, blah, but I love it. So plant fats tend to be more healthy, but they are liquid at room temperature, whereas animal fats are solid at room temperature. And in general, animal fats tend to taste better. So if you're going to, say, eat toast in the morning, a lot of people like to use butter on their toast because it tastes good. And not many people want to take out canola oil and spread it on their toast because it's liquid and it doesn't really have a good flavor. So in the 80s, scientists tried to figure out a way, it was like the end of the 70s, beginning of the 80s, they were trying to figure out a way to make liquid fats, so plant fats, solid. And we know that The main difference between the liquids and the solids is that the solids have no double bonds. They have more hydrogen. So they thought, what if we just add hydrogen then? So they hydrogenated the plant fats, making them solid. Once they had the solid plant fat, then they added some coloring so that it looked pretty, like a yellow color, so it looks like butter. And they added some flavoring to make it taste like butter. And all of a sudden they had this stuff that was equivalent to butter, but it was plant fat, so it was healthier, and it was called margarine. And everybody loved it, and it was good. Until we found out that it wasn't good, right? So we knew that saturated fats were bad. So animal fats can cause problems. Trans fats, we didn't know. So at the time, we thought they were fine. Now we know that they're super bad because not only do they increase bad cholesterol, the LDLs, but they also decrease good cholesterol. So they're worse than saturated fats. So now we don't use trans fats in anything really. There are in nature some trans fats, but if they're natural, we're not going to stop that. But in our foods, we don't use trans fats anymore. So another example of fats are phospholipids. Phospholipids are some of the most important lipids that we will talk about this semester, and the reason for that is because they make up the plasma membrane. So most of you have, well, no, I always say most of you because I teach a lot of upper level biology classes as well, and I'm assuming they've had general biology, but this is general biology, so you may not have heard of this. Phospholipids are structures, you can see one over here, that have a glycerol, and they have two fatty acid tails, okay? Phospholipids are amphipathic, so the glycerol head is going to interact with water. The fatty acid tails do not. And they make up the plasma membrane of our cells, and they help to basically act as a bouncer at a bar that allows certain people in and says, no, you're not getting in to others, right? So they're allowing certain molecules through the membrane and other molecules they keep out. Phospholipids they're amphipathic, I told you that, so they have a charged and an uncharged group. The charged group is called the hydrophilic head. So I told you what hydrophobic was. Hydrophilic means water, hydro again, and philic means loving. So if you love something, then you would be considered, well, I guess I've never really heard that, but just know the term philic means loving or desiring. So for these phospholipid heads, the glycerol, they are water-loving heads. So they're going to embrace the water and they can dissolve in water, whereas the tails are hydrophobic. And so this is what the plasma membrane looks like. It's a double membrane. So you have heads and tails here, you have heads and tails here. The heads face out towards the extracellular or intracellular environment. The tails are sandwiched in between. So if this was like the heads are the bread, these would be the meat of the bread. We also have steroids. Steroids are molecules that are based off of cholesterol. And so cholesterol is like their parent molecule. And then all steroid molecules are based off of that. And steroids tend to have the ability to interact with other molecules and cause changes. So estrogen, testosterone, progesterone, these are steroid molecules that can enter the body and they can cause changes. Testosterone helps in producing male sex cells called sperm. Estrogen helps in producing female sex cells called eggs, or activating, I should say. Estrogen and progesterone prepare the uterus for implantation. Testosterone also acts on muscles, helps in muscle building, helps to influence the secondary sexual characteristics of males, whereas estrogen helps influence the secondary sexual characteristics of females. Another example is vitamin D. Vitamin D is a steroid molecule that is initially started or produced by the skin and then continued in the liver and the kidneys. And it acts on blood calcium levels. I'm going to stop here and we'll finish up this chapter. Hold on a second. I'm going to pause it for a second. I'm back. I decided I'm not going to stop. I know this is already 21 minutes long, but I've only got about 10 slides left, so I'm just going to finish up right here, okay? If you need a break, pause me, and I will be here as soon as you are ready to listen to me ramble on about proteins, okay? So proteins are macromolecules that make up pretty much your entire body. So think about your hair. Touch your hair. Your hair is made of protein. It's made of keratin, which is a protein that's also found in our skin, and our skin is made up of protein. Muscles that allow us to do work are made of protein. Pigment that makes up our skin color, hair color, our eye color is made of protein. So we are proteinaceous organisms. We are basically made up of proteins. Proteins make us they give us all the structure and characteristics that we have, okay? Proteins are important in structural influence of the cells, but they also act as enzymes. An enzyme is a protein that increases reactions. So they increase the amount of reactions that occur in a given time. They can be hormones, which a hormone is a molecule that interacts with other molecules inside the body. They can protect our cells. So antibodies are protectors. And all proteins are made up of amino acids. So the monomers of proteins are amino acids. So this is a basic, very generic amino acid. All amino acids have the same amine group. They have a central carbon ring with a hydrogen on it, and then they have a carboxyl group. The thing that changes in all proteins is this R group. This R group is what makes each amino acid different. There are 20 different amino acids, and they can link together in multiple ways to make different proteins, all the proteins of our body. Protein structure is what determines its function. So proteins, when they fold up, are going to form a specific structure, and then that determines how they function. The primary structure of a protein is called a peptide or a polypeptide chain. So when amino acids bond together, they form peptide bonds through a process called dehydration synthesis. So remember me saying that before? All organic molecules are bonded together through dehydration synthesis. Once we have this polypeptide of amino acids, then the R groups, which can be charged or might be uncharged, are going to start folding. They're going to, so any regions that have a partial positive charge are going to be attracted to other regions that have negative charges, and they're going to be repelled by other positive charges. And so you're going to have this folding of the protein. That's known as the secondary structure. You can form either a beta pleated sheet or an alpha helix as the secondary structure. The tertiary structure is actually the structure of the protein. So once the protein folds into its final form, its tertiary structure, it is a fully developed protein. But some proteins actually have a quaternary structure where you have more than one polypeptide that folds into its tertiary structure together. They form what we call a quaternary structure. So basically you have more than one protein folding together. So here we have the primary structure, a bunch of amino acids that are bonded, and each of these bonds is called a peptide bond. Here's the secondary structure. Here's the alpha helix, and here's the beta pleated sheet. These are very weak. They're held together by weak hydrogen bonding and some other intermolecular interactions. Here's the tertiary structure. So this would be the fully functional shape of a protein. But then some proteins fold into their tertiary form in groups. So here's an example. This is hemoglobin, which has 4 proteins that folded together, forming this quaternary structure. So proteins are very important to the body's structure and for the body's function. And if we change the structure of the protein, then the function of that protein changes. So structure and function are super important. Denaturation is loss of protein structure. And when you lose the structure, you lose the function. So an example, if you've never done this, which I'm pretty sure every one of you have done this before, but you cook an egg. So you break the egg, you put it on a hot skillet, and you watch. Initially, you have this clear, viscous fluid, and then you have this yellow yolk. The clear viscous fluid is protein. And it's actually called albumin protein. When we break or when we cook it, we heat it up a lot. The clear fluid turns into a white, kind of like a rubbery substance, right? It feels kind of rubbery, semi-solid. That is the albumin protein still, but it's changed in structure, and so it no longer has the same function. So because of this, the human body, which is made up of proteins, has to be protected from major temperature fluctuations, because if we increase our temperature too much, we could actually break proteins down or denature them, and then they won't have the function. So body temperatures, when you have a fever, they get too high, they can actually start breaking down proteins in the brain, which can cause brain damage. The last macromolecule that we'll talk about is nucleic acid. So nucleic acids are what basically tell our cells to shape proteins. They're made up of nucleotides, and so there are 2 macromolecules, DNA and RNA, and then we always like to talk about ATP, which is a monomer, but it's a really important monomer. So nucleotides are made up of a nitrogen containing base, like this one, adenine, with a sugar and a phosphate. That's what a nucleotide is. So in DNA, we have 4 nucleotides, adenine, guanine, cytosine, and thymine. In RNA, there are 4 nucleotides, adenine, guanine, cytosine, and uracil. There's no thymine in RNA. So DNA is the instructions that make up proteins. So it's basically the instruction manual for our cells. In DNA, adenine and thymine always pair together. In DNA, cytosine and guanine always pair together. So we can actually identify the amount of the different molecules just by knowing one of them. If we know that our cells contain 25% adenine, then we know that our cells also must contain approximately 25% thymine because they always pair together. And then if we know that, that's 50% of all the DNA in our system. The other 50% has to be distributed equally between cytosine and guanine. DNA is always double-stranded, and it is made up of the sugar, or it's made with the sugar deoxyribose. So it's actually known as deoxyribonucleic acids. DNA runs anti-parallel, so you can see the structure of DNA here. A always pairs with T, G with C. The backbone is here. It's the sugar phosphate backbone, and then these are the rungs of the ladder. If this one's running in this direction, the other one will be running in this direction, the opposite direction, okay? So we have one going upwards, one going downwards, but they're still going to be paired properly. RNA then is what makes up, or I shouldn't say that, ribonucleic acid is made up of RNA monomers, adenine, uracil, cytosine, and guanine. RNA is single-stranded and it has the sugar ribose. So RNA is what carries out the job of DNA. So DNA kind of sits back and carries the instructions and tells RNA what to do. RNA actually is the worker. So you'd be like the boss, the architect maybe, that's in the room drawing up the plans, and then RNA would be the workers that actually make the building. Make sense? The last nucleic acid that I like to talk about is ATP. ATP is a nucleotide, but it's formed from an adenine, a ribose, and 3 phosphate groups. So adenosine triphosphate. And this is the energy that all cells use. So ATP is super important in producing energy that we need for all of our different metabolic reactions in life. ATP can be broken down, and when it is, it loses a phosphate. So if we take one phosphate off, then we produce ADP, adenosine diphosphate, so we only have 2 phosphates attached. And the energy released runs different metabolic reactions. Then we can always add a phosphate to the ADP, adenosine diphosphate, producing the adenosine triphosphate, or ATP again, when we want to make more energy. So here we have ATP, high energy chemical bond. We break that bond, producing ADP plus the phosphate, and energy is released. All right, I am done. This was a 30-minute video. I am super sorry, but actually, no, I'm not, because I feel like it's okay. You can always stop and restart the video when you're ready. So you have a wonderful day and enjoy the rest of your weekend. And you can listen to me again whenever you feel like it. Bye.